This is my conversation with Dr. Peter Dedon. Pete helped create the Department of Biological Engineering at MIT and also started the Dedon Lab in Singapore. Our discussion covers everything from infectious disease and antimicrobial resistance to gene editing, stem cell research, and the pharmaceutical industry. I found our conversation to be extremely valuable and insightful, and I hope that you do too. All right. Well, to get started, can you talk a little high level about the research that you do and your lab in Singapore?
Absolutely. Yeah, the research I do both at MIT and in Singapore uh, kind of comes from my roots. I went to medical school, didn't do any clinical training beyond that, so I'm not licensed to practice medicine. But I do have a PhD in pharmacology and drug development. Uh, Technically, I'm a nucleic acid chemist, DNA and RNA chemist at heart, and everything we do in the lab stems from that. And my involvement in Singapore was a bit of serendipity that got me into the infectious disease realm about 15 years ago. It was an accident where a colleague who was in this SMART program in Singapore that MIT has uh, invited me to come and explore and see if I would be interested in bringing my technology into the program. And sure enough, I got there, got bit by the bug, and ended up now 15 years in. I'm leading the Antimicrobial Resistance IRG. That's 8 MIT professors and about 8 or 9 professors from different universities in Singapore, Nanyang Technological University, the National University of Singapore, Duke NUS Medical School. And we're all doing different things related to antimicrobial resistance and infectious disease. I'm in the drug development side. We have quite a few of us doing small molecule development, biologics, antibodies that were hot in the news with COVID. We've got people making diagnostic kits. One of them is currently in Singapore being developed for use, for example, at the airport, where you want a really quick, fast test done right on the spot. Another
Hmm.
group was doing sewage surveillance, which sounds creepy, but by sampling the sewers in neighborhoods, you can check for outbreaks within small areas of places like Singapore. They were doing this in Dubai, doing it in Cambridge, Massachusetts, and that became a huge part of uh, 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 public health efforts during the pandemic, trying to figure out where the outbreaks are happening to help prevent further spread. So that's the practical stuff we do. We do translational science there. A lot of basic stuff, but then we translate it into the community, public health, or to start companies, to make new products for public health, all those realms. So that's kind of the work that I do in a nutshell.
Very cool. So, how much of a shock or lack thereof was COVID in the pandemic in the first place? Because I know, like Bill Gates, for example, gave a TED talk years before, and a number of, I'm sure, health professionals kind of saw the writing on the wall. Um, what was that like from your side and from the world that you kind of work in?
Yeah, yeah. I think as scientists, biomedical scientists, and infectious disease scientists, especially, you knew it was coming. You just didn't know when. Uh, Ebola should have been the scare for that one, with people flying on planes back to the US from Ebola hotspots. Uh, and that Ebola is tragic as it is, it's self limiting. It
Mm,
People die very quickly from
right.
this. The pandemic was the ultimate wake up call of something that is indolent and then explodes, but doesn't kill everybody in sight. So we're
Hmm.
still a little bit lethargic about taking action, even when we know there's a virus spreading. My history with the pandemic was I was in Singapore right before the Lunar New Year in 2020, and had taken my group out to a hotel. They have these very fancy buffets at hotels in Singapore, really a lot of fun. And I took my group out there for a bit of a party. We like to do that once a year for some socializing and fun. Got a
Hmm.
call the next day from the hotel management saying, "Hey, there was a group of tourists from Wuhan in the party room next to yours, and they had that virus." And at the time it was like, "Oh, well, that's interesting. Gosh, I hope we don't get it." I flew back on a plane 2 days later to the United States, immediately developed the flu. 2 weeks of feeling like terrible. No lung stuff. Colleague of mine in Singapore had bad pneumonia at exactly the same time. We all conclude now after the fact that we had COVID, and I
Mm-hmm.
probably brought it back with me. I'm not the index case because it arrived in the US long before, but that was the door that opened us to the pandemic. Now, I
Mm-hmm.
will say that our group, which of course immediately shut down, the whole world went into the big stop then, right, for the pandemic.
Mm-hmm.
One of the things that our group did was to immediately pivot from all the work we were doing on antimicrobial resistance to doing COVID immediately. The teams made, one group made an antibody against COVID that got into phase 3 clinical trials. And they made that antibody within 3 months of
Wow.
getting the first viral epitope. And it's still the most promising antibody, although with the pandemic sort of being over, so to speak, the development has slowed.
Mm-hmm.
The sewage surveillance group immediately started doing sewage surveillance around Singapore and helped stem further spread. The diagnostics team had a diagnostic kit ready to go within a couple of months, and they were testing it on people. I actually went back to Singapore and within like 6 months, right in the heart of the pandemic, stayed in the hotel for 2 weeks quarantine,
Mm-hmm.
and then went back. I actually got my blood drawn to help test that kit. So our team pivoted rapidly to do this. That's what's coming for the future now, is when research groups at universities and in academic centers and in industry can pivot quickly to the
Mm.
next pandemic that comes, bacterial,
Sure.
viral. And I think that's the secret to the future, is how fast can we respond? And boy, look at the response that Moderna, Pfizer BioNTech had with messenger RNA vaccines within months of the outbreak. That's, in my 67 years of life, that's the most incredible technological breakthrough that I've seen.
Sure. How do you think we can handle like efficient and sufficient testing of, these kind of extreme case necessary developments? Like I know one of the big concerns, at least in the beginning with all the vaccines and stuff, was, how long has this all been tested? Has this been tested effectively in humans and all that kind of stuff? How do you think we balance that with, um, like the experimental drugs and sort of releasing that stuff when it's medically necessary during pandemics?
That's always a tough call, always. And it's a risk filled call. When the first vaccines came out, they were limited to the most necessary cases. And what were the most necessary cases? People with uh, other illnesses that made them
Right.
highly susceptible to being
Comorbidity
killed.
factors. Yeah.
Yes, exactly. The elderly. They didn't give it to kids and babies because of the risk, at that moment might have been too high, might have caused some toxicity. And so all of drug development is balancing safety and efficacy. Does it work and is it safe? Those are the only two things you ever have to be concerned about. And you put safety first until you've got a life threatening situation. So cancer drugs are very toxic, typically. They have been in the past. The modern ones are a little less toxic now.
Hmm.
So you really want to do your clinical trials only in the people for whom this is the last hope they've got. And then you go back, and if it's safe, relatively safe, you can go back and you tolerate a lot of toxicity for a case where somebody's simply going to die if they don't get something. Well, messenger RNA vaccines for 1 billion people, we're not going to tolerate a lot of toxicity. Vaccines in general are very difficult to make because everybody has to react well and it has to be safe. So
Hmm.
the testing they did in animals early on showed it was going to work. Did we rush to get it in? Well, yeah, because a lot of people were dying. And so you do have that emergency overlay that justifies a bit of risk. And it paid off. All the fuss now about long COVID, the reaction to vaccines, cardiovascular complications of vaccines, they're extremely rare compared to other drugs that are already on the market. and that we tolerate those risks because of the need for that drug.
More broadly speaking, do you think that in the US at least the way pharma companies are structured in terms of like the peer review and the testing and incentivizing, profit over certain other things, do you see concerns with that in terms of safety and some of these uh, factors that you're sort of talking about? Or do you think the sort of system we have is as good as it's going to get? Because I mean, historically, right? It's obviously hindsight 2020, but it's easy to see certain drugs that came out that obviously were either less effective or were dangerous at the time, and then we like to point fingers and say, oh well, that's a really bad thing. Like how could Pfizer just put this out and then let this harm people? But at the same time, how much of that is rooted in just lack of perfect information at the time versus how much of that is rooted in poor incentives and corner cutting? That's something I'm curious about, if you've seen any of that, or what your opinion is on kind of the landscape of big pharma.
Oh, that's a complicated one. Yeah, the nothing's perfect in the world of drug discovery and development. Big pharma is, you have to report to the owners of the company. You have to turn a profit. It's a profit-making enterprise. How big that profit is, that's not my judgment. That's not my call. You know, I would love to start a drug company that is nonprofit. I can start a company if I can have good salaries for people, and they can have a good retirement plan, and it's secure jobs and it's good jobs, and make drugs that get on the market and they make zero profit except to pay back the costs of making that drug. I would love to do that. I would really love to do that. That's a really hard one that nobody's succeeded in doing yet. Are there other business models for developing things like infectious disease treating drugs? Yes, there are, and they're emerging now. They're finally, we're coming to the conclusion after all the big pharma has abandoned antimicrobials, antibiotics, all of that stuff. There's no profit in it because a lot of that is not profitable. We're going to put some of those new drugs and hold them until we absolutely need them, because the antimicrobial resistance threat is growing. We want to keep some drugs in reserve so a urinary tract infection isn't killing normal, healthy, otherwise healthy people. So how do you pay for a drug that you're putting in reserve and not selling? Okay, we got to figure out a way to manage that. Government's got to help. Foundations have to help. Pharma has to chip in somewhere, and we'll figure out a model. To your point, the pandemic was this crazy explosion of noise when it comes to claims of Donald Trump saying we can drink Drano, and people testing repurposing drugs. Now, some of the
Right.
repurposing really worked, and it came out ahead, but we had people doing repurposing vitamins to say, oh, they're fighting COVID. Well, we rushed in a lot there. And once the dust settled and you look at the data and scientists scrutinize it, the crap was put into the box of crap, and the stuff that actually worked was moved forward for further testing. So yes, there's a rush. Now, was that profit driven? Okay, these are human beings doing this. And who's out repurposing drugs? Somebody who wants to make $1 billion with a drug that's going to fight pandemics. Uh, I can't justify any profit driven motive in a pandemic or a crisis. It really is all about, let's put all the oars in the water and make something that
Mm-hmm.
saves millions of lives. Okay. Profit motives, you can hear me kind of wandering all over the place here. It's a tough area.
Of course,
Everybody's
yeah.
bashing big pharma, but if you really look at the cost of developing the drug, like the cost of lab coats and safety glasses for workers and pipettes to do the experiments, it's $1 billion from concept to market,
Mm-hmm.
just the cost.
Right.
Now, if you want to make a profit on that, you've got to add to that cost. And then that depends on how many people are going to buy this drug. For a pandemic, for COVID, it was billions, right? So all of a sudden, you divide the cost took to make the drug by how many times you're going to sell it. That's the cost per dose. Well, there's a
Mm-hmm.
profit margin built in there too, right? Fine, we can fuss about the profit margin, but it still cost you $1 billion to develop that drug.
Mm-hmm.
And you got to make that back if you're just going to break even. Did we break even? Well, no, because the government paid for a lot of that cost up front,
Sure.
right? So we as the public got great medicines we got it for free in the US. And who paid for that? Well, our tax dollars did to a large extent. Now, moving forward, the drug companies now know how to make these. They've de-risked the manufacturing process. It's going to cost less. Government won't have to step in necessarily.
Sure.
So, because like flu vaccine is all made commercially and it's very cheap.
Mm-hmm.
So we'll get there, I think. Pharma's going to get squeezed as we move forward with insurance costs and everything else, and we have to evolve and adapt and figure out new business models. But I'm an optimist. I'm a glass half full person. I think we're going to get there.
For sure. For sure. And I think, at least from my perspective, I can certainly see the gray area and some not great things that have happened in industry over the years, just looking backwards. But at the same time, I also don't think we would have a lot of the drugs and the medical advances that we do have if we didn't have, a system where there was a fair incentive for development. But that brings me to another question of mine. So you had mentioned your big concern with antibiotic resistance to ordinary infection. Um, how big is that danger compared to things like heart disease, obesity, cancer, things like that? And why exactly? I know you touched on this a little bit, but why exactly are we not seeing sufficient development of these new antibiotics at pharma companies today?
So antimicrobial resistance is growing exponentially. It's exploding partly because bugs evolve to adapt to the antibiotics we're currently taking. Humans mutate and evolve very slowly. Bugs do it in hours. And bugs pick up genes from other bugs and they adapt and they survive. And so it's, every time we take an antibiotic for any infection we've got, we're selecting for drug resistant strains somewhere, somehow. You take an antibiotic, you put it in your mouth, it moves around your body, it goes into the urine, it goes into the feces, that ends up in the sewer system. That sewer system ends up in the river, and that ends up flowing around the world in the ocean. Okay, that's not an outrageous statement to say the presence of those antibiotics in the environment are again helping to select for the resistant strains, which will then get back to people. We
Sure.
deliver it to animals in agriculture. And the meat, the produce, the things we eat contain antibiotic residues. The whole new modern world is focused on shepherding antibiotics, being very careful not to give them out for everybody that comes in with a fever, or to give them out when we think it's either a viral infection where an antibiotic doesn't work. We need another drug for that.
Mm-hmm.
So to your point, it's evolving quickly, and it hasn't quite reached the stage where the general public is banging on doors trying to solve the problem. And that's where the public is often slow to see the
Mm-hmm.
crisis as the
Just like
house is
with
on fire
COVID in the
down the
beginning of
street,
COVID.
just like COVID. Uh, and so, unfortunately, we're going to reach a crisis point where public demand reinvests money from the government, reinvests money from pharma to push to get this done. It's happening. There's a lot of groups like mine. There's groups all over the place. There are now foundations set up to get money to put millions and millions into antimicrobial development because pharma has abandoned it because it is not profitable. So there we got to come up with that business model. Let's figure out a way to make them at least break even. But then you have to consider the fact that the cost of $1 billion drug in the US, how can you afford that in a country where the average income is $1,000 a year? At $1,000 a dose, I'm sorry, you cannot do that. We need a new global health business model for getting antimicrobials into the world. Why? Because if we're all taking wonderful antibiotics here in the US, everybody else is taking the old antibiotics, generating the resistance, and that's eventually going to come to the rich countries. So there's a motivation and an incentive for the whole world to have some care and access to antimicrobials, better shepherding to prevent resistance, try to reduce it, but it's here and it's not going away.
How soon do you think we're going to start seeing like UTIs and no, everyday infections that become hard to treat?
Oh, it's here. It's here now. And it's been here for a long time. I had a scientist in my lab 6, 7 years ago whose wife had a normal urinary tract infection. She was in the hospital for a couple of weeks after several trials of different antibiotics from the doctor's office all failed, and the infection got worse. And finally, they found an antibiotic that worked. Sorry, that's antimicrobial resistance. And that was an
Wow.
ordinary urinary tract infection. It's now worse. E. coli, Escherichia coli, is the main bacteria that causes urinary tract infections. There's a huge increase in drug resistant E. coli. It's now on uh, the Centers for Disease Control, like most wanted list, along with several other really bad things. There's this tuberculosis caused by Mycobacterium tuberculosis. There's now a strain that is completely resistant to all antimicrobials. It's called extreme, I think it's called extreme tuberculosis. I forget what the exact acronym is, but we had a case in the US. It's here. Yeah. But yet it's not frequent enough to make the New York Times front page every week. And the public is not aware of it yet, but
Jeez.
it's here.
D- do you have any ideas in terms of how to make the venture of new drug development in that field more profitable or more feasible?
This is what people in my position and investigators at universities, public health officials are all working toward. We had a symposium here at the beginning of July with quite a few people from my Singapore group. And we had some headline speakers. Andrew Lowe, who looks at the pharmacoeconomics. What are the economics of drug discovery and development? Well, they're miserable. Okay, we've had people on the front lines in the clinic trying to develop the drugs. It's tough, but you put all this brain power together, you come up with new, you get business people involved, and you start to have these teams figuring out ways to make new drugs. There's several programs now in the UX, one called CarbX, that bring a ton of money together from different sources, drug companies, government, foundations, and they pick the most promising candidates for antimicrobials and they push them into development in ways that professors can't do, in ways that drug companies don't want to do. And so they're filling this gap, this valley of death, if you will, where we can finally get drugs into the clinic. Most drugs that go in the clinic don't succeed because drug development is really tough. You put a new molecule into a human being, humans may not react the way mice do, and it may prove to be too toxic. Okay, well, we can't just give toxic drugs to everybody. It's got to be safe and it's got to be effective. So, what is happening now to solve the problem of no antimicrobials? There's a ton of people working 24/7 to figure out how to do this. Now we need the business models. Now we need to figure out then how to push them forward faster. There is an uptick in the number of new antimicrobials, antibiotics getting on the market. We need more. Ear infections in kids. Okay, talk about a commonplace market, right? Uh, a lot of the bugs that cause those ear infections are now drug resistant. Well, fortunately, we've still got some drugs that work, but let's look forward to the horizon where we don't want our kids dying from ear infections. We're not going back to the 1820s where one kid in a family always died from some infection. Sorry, not going back that way.
Mm-hmm. Wow, that's an intense
Yeah, sorry, I
conversation
don't
to
mean to be
have.
a doom and gloom here, Mr. Cold Bucket of Water, but
No, no, I think it's important to have, and that's part of the reason I've been trying to have, like, conversations like this myself, and I mean, educating both myself and just I think the more information in general that's out there and available in a lot of different fields are just valuable to sort of spread awareness. Do you think some of the technology advancements like AI and the large learning models and that kind of stuff has the capacity to significantly cut costs of some of this development? Or do you think this is nuanced enough that it's going to be tough to sort of apply any of this tech, at least in the short term, in from an economic perspective?
Yeah, that's a really good question. And there's a lot of activity now to cut that $1 billion I said earlier, try to cut that down to 200 million. Let set realistic goals. Let's start setting milestones to cut the cost. How do you do that? Drug discovery and development is a 10-year process. It starts, first of all, you have to find the target for the drug in the organism. What are you going after? How do you then choose a molecule that attacks that target, kills it, and then kills the bug? How do you take that molecule and make it safe? Change the structure a little bit. Change the structure so it's potent in a human, uh, so that you get a drug that's safe and effective in a human. Once you get to that, how do you make enough of it, synthesize it to run a clinical trial? How do you synthesize tons, literally tons of that drug to put it on the market and get it into vials and get it into doctors' offices?
Hmm.
Okay. All of those steps take time and have to be developed. They call it the CMC phase of drug discovery and development. That's chemistry and manufacturing processes. How do you manufacture tons, put it in little bottles? Is it stable? How do you make it stable on the shelf so that people can actually go to the pharmacist, get the pill, and pop it, right? There's people working on every single one of those steps. And I'll give you an example from one of my colleagues who makes antibodies, biologics, against infectious disease. It used to be that it would take 5, 6, 7 years to go from identifying the protein on the bacteria or the virus that you want to attack to getting the antibody into people, into clinical trials.
Hmm.
He progressively shortened that time frame so that with the COVID antibody, between the time they identified the target to the time it was ready to put into people, 3 months.
Wow.
So you go from 48 months down to 3 months. You've cut costs dramatically. Think of the companies that have to keep staff on payroll, and they're not doing anything, but they've got to be there for each step of the way. You shorten that development phase like that. That's crazy in terms of the reduction in costs. The Food and Drug Administration requires a whole series of tests to prove safety. Well, we've now got technologies where we can take several of these tests, put them into one measurement. It's called omics science. I can measure thousands of molecules all at once, and I can then do all those assays in one. If I can cut the time frame for testing the safety of the drug down from years down to months, a couple of months, boom, another huge cost savings.
Right.
All this stuff, how do you find the target and find the molecule? We now screen millions of compounds on the computer using AI to see if they fit the target and get candidates that we can then make and test in the test tube. Boom, cut that search process down by years.
Right.
Okay, let's work on each of these steps, and pretty soon we're cutting that billion down to millions. And once it's down to millions, your cost per dose is way down, super down. So,
So it's
yeah,
lower the
it's
barrier to
happening.
entry for small startups and groups essentially is the biggest kind of approach.
Make it more profitable and make it more affordable so that you got both worlds, the business world and the public health world, getting the best of things, getting the drugs they need to treat whatever's the problem going on.
Sure. So I don't know if this is just my if this makes sense as a question or not. It might be my lack of knowledge in the field. But how does technologies like CRISPR and things that allow you to sort of edit and like genetic information and gene expression, does that like have any place in this discussion, or is that like kind of a completely separate topic.
No, that all these technologies, modern gene editing technologies, modern omic science, modern computational science, all of these things are important. I'll use the CRISPR example. We can now use CRISPR to make a library of human cells where we knock out or knock in specific genes, right? And now we can go test for which of these genes is important for a cell to respond to a therapeutic. And we couldn't do that before. You had to make one at a time, test them one at a time. There's 20,000 genes in a human. How do you do that one at a time? Let's do them all right now with CRISPR. And we can make these massive libraries, and they call it massively parallel screening. So CRISPR is a vital technology to be able to tackle some of the molecular cellular mechanisms of discovering targets, making antibiotics. You still have to go into animals. There's no way we're ever going to get rid of animal testing to see that we're going to get into a human. Not in my lifetime, because it's got to be safe and efficacious. So more technology for animal alternatives, for shortening the number of years we need to go into animals.
Right.
So CRISPR can do that. All the mice that we can make now, engineered mice, where we can use CRISPR to knock in a target or knock out a target and make animal models that shorten the timeline to development. Oh, it's very important stuff.
With technologies like CRISPR and gene editing, how far along are we in that development? Like I know, like from what I've heard, we can edit genes effectively in some capacity, but in terms of at scale and for humans, um, how far away is that?
That's a ways away. From my perspective of a therapeutic for people that's safe and effective, let's say things are already happening. There's gene editing drugs in development. We've had tragedies where people have died and there's setbacks from the trials of these things.
Mm-hmm.
We still don't know fully the consequences of CRISPR technology. It isn't perfect, and that's the problem. Sometimes it makes a mistake and puts things in the wrong place. What's the risk of that? Well, maybe it's not much, but then maybe somebody dies. Okay, so we've got a lot to work out before this becomes. Sorry about that. I should have done that before getting on here.
No worries.
We're a long way before this becomes something like a statin for lowering your cholesterol, where there's 12 of them, and they're all on the market, and millions take them, and they're safe. Long way from that. But experimentally, for the worst case scenarios of fatal diseases, where if we don't get it, kids are going to die, adults are going to die, we'll tolerate the risks. We're getting to that point very quickly. So we're at that crossroads, if you will, or
Right. There's this
yeah.
there's this sort of gray area with a lot of the ethics of, modifying genes and all of that kind of stuff too, that is probably pretty tricky to navigate, I would imagine.
Very tricky. So I teach a drug discovery and development class yeah, twice a year with this colleague of mine who shortened the antibody production timeline. Uh, we have built into that discussions of the ethics of drug discovery and development. Big time importance. And the students and young people are picking up on this in terms of like the cost of global medications for global infectious disease. There's the new generation of students who are now going to populate the drug companies, who are now going to start the companies, are growing up with this sensibility for ethics and global health. And so you're now going to have this dynamics going on to actually move forward with making drugs that cost a penny a dose. How can we develop the technology to reduce the cost of making a drug? So to your point of the ethics and all this of gene therapy, yeah, there's some sticky wickets there to work out.
For sure. So, kind of shifting gears a little bit, I did want to bring up some epigenetic related questions that I have. How much really does behavior and environment influence gene expression compared to your pure genetics? And as time is going on, are we finding out kind of the tide is shifting more towards, oh, it's even more your environment, it's even more your behavior, or is it shifting the other way of, well, we're finding out that like, genetics are sort of a bigger role than we thought at first?
Yeah. Nobody's ever going to answer that question because every time we get a new answer, we come up with 10 more questions that raise doubts on what we presumed previously. Gene and environment uh, it is so complicated. It is no longer just gene and environment, right? 20,000 genes in humans. What what schedules the turning on and turning off of all those genes? Okay, in the past, in old days, we would say, oh, it's transcription factors that turn on all the genes. Well, what turned on the transcription factors? That's where you're talking about epigenomes now, where there are changes in the DNA, the genetic material in every cell, chemical modifications that schedule and turn on a gene when it's needed. So there's a whole system in place to change these epigenetic marks on genes as we go from fertilized egg all the way to 67-year-old cranky old man, right? These are constantly evolving, shifting as we develop as human beings. The scheduling of that transcription of those genes is done by epigenetics. I'm in a field now called the epitranscriptome, where there's modifications of the RNA products of genes that then go get turned into the protein building blocks of cells. Well, the scheduling of the production of the protein building blocks is now something that is not controlled necessarily by the genome. There's a whole system of regulating modifications of the RNA that
Mm-hmm.
regulate how quickly, how much, and when proteins get made. That's complicated. You got epigenomes and epitranscriptomes doing the scheduling. You have a genome that is what the end products will be. But what determines now the epitranscriptome and epigenome activity? We're just working that out. I think the
Right.
microbiome is the perfect example where we don't know yet how much environment and genes play in determining us. The microbiome, the bacteria, let's say in your guts,
Mm-hmm.
are absolutely critical to your existence. This idea of washing your hands every 15 minutes, don't do that. I'm sorry, we need some bacteria. They're the bad ones. Yes, wash your hands after, you go to the bathroom and after you cough in your hands, fine. But we need these bacteria. They make molecules that are like vitamins for our body. There's something called cuine, which is the precursor to an RNA modification I study. Humans can't make the cuine, bacteria can. You get it from your diet or your bacteria. You need that cuine in all the cells of your body. And in fact, there's studies that show that if you lack cuine, there are a huge number of psychiatric mental disorders that arise, schizophrenia. I'm sorry, that is wicked complicated. But it tells you that you have this set of bacteria, and in each human being on the planet, the spectrum of bacteria in the gut is a little different.
Mm-hmm.
So is that environment or is that our genetics that determine that set of bacteria? It's both. It's what we eat, it's the environment we're in, it's our family, but it's also our genetic makeup for the activity of the white blood cells in your gut that kill off bacteria. So there's a place where is that an environmental thing or is that like an organ in our body now? That's just mind blowing, complicated to think of how what we eat can affect our microbiome, that affects our health in ways that our genome is not, you can't mine the genome with AI and predict that. Sorry,
Mm-hmm.
there are some things we can't predict by simply looking at our genome or looking at our environment. Yeah.
Yeah,
Sorry,
that's
I just made it 10 times more complicated, but for me as an explorer in this area, it's wicked complicated.
sure, sure. And I think that uh that's one thing that I'm curious to see if quantum computing can like start to in the coming decades start to address some of these challenges, just because it's sort of built to um, be able to model things that have these complex interactions very, on the same basis that our actual environment exists. So it'll be very interesting to see. But I agree. I think like trying to I mean, ultimately, what we're trying to do, right, is create non-complete scenarios and use those to explain the complete picture. And it's going to be a long time and a lot of probably different steps forward and steps backward before we really make significant progress in that area. So one thing I'm curious about
Yeah.
too is, yeah, were you going to add
Oh
something?
no, I was just going. I was going to agree with what you're saying, and step back for a minute. And I just sort of belittled the role of AI in and computational work in predicting genome function.
Mm-hmm.
I'll go back and say now that machine learning, which is the basis for AI, all of those tools are absolutely essential now to do exactly what you said. Take all this omic data that I can accumulate with my machines and compute that and develop that into a model. Give me a prediction of where that's going to go. I can go back in the cell or the mouse and I can test that prediction.
Mm-hmm.
And that kind of learning is exploding. It's exponential. When I started in graduate studies, a kilobyte of information was a lot on a little floppy disk, right? Now we're talking petabytes, gajillion amounts of information. We need new hardware to store the data. And so as the technologies evolve, all of these things are going to come to play, and maybe we will get models of the microbiome and predictive power for how we can regulate it now as a tool for therapeutics, for understanding human biology and health. So you're absolutely right. Everything's evolving at the same time, and it's now exponential in terms of the information that we're getting out of all of this.
Mm-hmm. um, I wanted to bring up, do you think that we're creating any dangers in our environment that we should be aware of? I mean, obviously, setting aside like the uh, complex, molecules and things we're dropping in the environments and everything with antibiotics and whatnot. Um, but like with things like plastics and that type of stuff, those inert compounds, do you think that there's concern with that stuff and how we kind of interact with that as biological entities? Or do you think that all of that is mostly overblown and not something to be super concerned about?
I started my career in carcinogenesis and toxicology, and that is how do chemicals in the environment, how do chemicals in your body damage the genetic material and cause cancer? So I have a sensitivity to these environmental questions. Also, I'm a member of the Center for Environmental Health Sciences at MIT. Uh, so that's a hard one. You can take the plastics thing that you just mentioned. All the plastics environment, we're now finding that it isn't just dolphins getting their noses caught in six-pack rings, right? That's the environmental hazard of plastics. They degrade into tiny little micro and nanoparticles.
Mm-hmm.
And those things we've never studied before, but they're turning out as they get into fish, as they get into plants, as they get into humans potentially. We don't know the effects of those things, toxic or otherwise. Um, and we look at nanoparticles, what we're learning now about microparticles and nanoparticles in our lungs from breathing air pollution. What are they going to do as you eat them and they go around to your cells? We know nanoparticles, many of them freely cross the membranes of your cells and get into your tissues and inside the cell. Okay, that's a little scary. All right. Now, am I going to be an alarmist and say that everything is horrible in life and we should just, fly to another planet where it's cleaner? No, I don't know the toxic effects of these things. That's why people are studying them. One of the things I would advocate for is funding for the research to investigate these things. That's where we really need advocacy to try to predict what is going to be the toxicity, the health effects of a lot of these environmental problems that we face. Yes, years ago it was DDT and all these long living chemicals in the environment now that stay in the body forever. What effect, as we learn, we accumulate information, we are learning from that. Some things we need to pay attention to more than others. because we just have
Hmm.
finite bandwidth and finite money to do anything, right? So
Hmm.
let's pick and choose our problems carefully, but yet put enough money aside for the basic researchers to go investigate some of these mechanisms. Are they important or are they not?
Yeah, that's another one I think is tough from an incentive perspective, right? Because a lot of the emitters of these particles and nanoparticles have no incentive to figure out necessarily if it's harming people other than the goodness of society and that kind of thing. And until we discover gen genuine dangers, I also have a hard time seeing the profitability of some of that research. um, so it kind of gets back to that same sort of idea of like, how do you incentivize something when the sort of benefit that's associated with the cost is probably down the road a little bit, which, as we see, makes things pretty complex.
It's that balance, right, between the current producers that have a financial incentive to keep producing the plastics that people are concerned about.
Hmm.
At the same time, I think we're in an age where young scientists, maybe old scientists, are motivated to go start companies, develop the technology that makes a plastic free environment feasible, makes a different kind of plastic that doesn't form these nanoparticles. You know, develop the technologies that solve that problem. It's going to take 10 years to do that. But I think there's a lot more of that kind of thinking going on now, especially among young students and researchers, to see what the problem is. Let's go make a technology to solve it. Let's go study it and figure out a way to solve it that doesn't punish people. I mean, the government can set regulations and has to on certain things, and that's
Hmm.
painful. There's politics in that, right? At the same time, let's look at the glass half full approach and say, let's go solve that problem. You look at solar power, how it used to be so financially unfeasible. It's all over the place now. It's financially feasible to have solar power on your house in many parts of the world. And look at the cost cutting where people are actually selling electricity back into the grid
Hmm.
from this. Okay, 20 years ago that was silly, and the power companies were fighting that. Okay,
Right.
well now maybe not. Maybe the business will evolve to see the financial benefits of a greener world in terms of look at electric cars, shifting from fossil fuel to electricity. Well, things are slow, but again, I'm the optimist to say I think there are ways for us to structure the world, to structure businesses,
Right.
and
Right.
incentives for young people to get out there and start companies to solve the problems.
Right. Yeah, the cars one is a little bit of a touchy subject for me because I'm a car guy and I don't want to say goodbye to my gas
No,
powered vehicles and the motors. But
yeah,
definitely, yeah. I mean, even just like taking Tesla for an example, that's obviously the easy example. But I mean, 20 years ago, no one would have even thought it was feasible to make a production kind of electric car. You'd run out of range, you wouldn't be able to charge it. And I mean, now even some of the technology that's in there in terms of like mapping routes and set, you know, optimizing for placement of superchargers and things like that is really amazing. And I think that'll be a while before we get over more to electric vehicles. Because we got the rest of the world that's still on gas vehicles, and even in the US
yeah.
it's going to be probably and past my lifetime when gas vehicles are gone. um, but it is pretty amazing to see just stuff like that, complete shifts. And some of these shifts you don't even see coming, or you don't even kind of realize them at the time that they're happening, and then it's just completely gone or completely different. One example that's pretty small, but I like to kind of remind myself is like headphones, for example. Like when I was a kid, I remember even into middle school, high school, it's like everybody had the headphones, and there'd be all the posts online about, oh, when your headphones go in your pocket and they get all tangled up and stuff. And now it's like I can't even tell you the last time I like saw someone out walking with wired headphones or earbuds. It's like
Well, and
crazy.
I'm wearing my earbuds right now, so yeah, here
Right,
we are.
right. So I mean, it's crazy. Like some stuff seems like it would be um like a long time coming, and it just kind of is there. And then other stuff, we see and we think is going to be different, and then it just takes forever. Um, so that is another thing. Like, as much as it's fascinating to talk about this stuff, like, there is an aspect of it where we just don't know, right? And one of my other favorite examples is like in the movie Blade Runner, the original one. They have all these flying cars and AIs that have, insane ability to just answer any question you have, which we're almost getting to that point. But then they would have the TVs that are, you know a foot thick, and incandescent like screens and stuff, and they had no idea, they couldn't even conceptualize the idea that we would have, ultra thin LED flat screen TVs and stuff. So it's pretty cool to look back and see kind of what we got wrong in our predictions, and what we got right. uh
Yeah.
I did want to ask, one other main topic was like stem cells and stem cell
Mhm.
injection and research and stuff like that. I'm not very educated in that, candidly, but what role does that play, and how has that been developing?
I too am not an expert in stem cell biology, but having been in this environment of biomedical research for many years, I see the potentials there. You know, the idea of taking a fundamental cell that can divide and evolve and grow into a mature tissue, a type of tissue. We got 30 plus organs in the human body, and dozens and dozens of different kinds of tissue, and trillions of cells that make those tissues. So in each of those tissues, there are these fundamental cell types that, as the mature cells die, which they always do, that's just life. Cells wear out after a couple of decades, and they die. And your brain cells don't replicate and replace as frequently as others. So that's what we call dementia and aging, is when enough cells die in your brain that you stop functioning. Okay, the
Hmm.
stem cells renew the mature cells and divide regularly. Now, can we generate new heart tissue, muscle tissue, brain tissue by injecting some of these stem cells and letting them grow in the environment of the organ and repopulate the mature functioning cells of that tissue? That's the key. Well, like gene therapy, this is sort of early stage stuff in terms of
Hmm.
how do you control it. What if you throw a stem cell in and it grows into this giant ball of useless tissue that is like a cancer? That is what cancer is. It just crowds out all the normal cells, and you stop the function of whatever organ it's packing. So, uh, you know, how do we prevent that? How do we predict it? How do we control it? How do we regulate it? That's all the stuff of current research. Artificial organs. Can I grow a human heart in a test tube and put it beating into a person, and thus not have to rely on transplants of dead people's hearts uh, to save lives? Well, that's the goal. I think it's quite far downstream. Now, there are organoids. They call them organoids. They're fake organs that people are now using in the drug industry, for example. We can make kind of a semi fake liver.
For
Most of the
testing.
drugs that you take in your body get metabolized in the liver. So in the drug industry now, we can have liver cells growing in culture that are getting more sophisticated because of things like stem cells. We can make fake organs, fake livers, that we can now put the drug in and get a realistic metabolism just like in a human. We're close to that. So we're getting there. Stem cells play a role in that kind of science and engineering. But we're still, nobody's squirting any stem cells in me for a little while. I got to see, I want to see the proof. I want to see the evidence. But it's
Sure,
exciting,
sure.
though, from the standpoint of the promise that it holds.
Yeah, for sure. It sounds like all these sort of different areas kind of overlap in different ways, and probably finding the best health outcomes for the most amount of people over time is going to be some combination of all of these things together, and shortening some of the testing phases, and improving the accuracy with stem cells, and then in other ways with some of the finding new ways to profitize some of this stuff will incentivize companies to do that, and all these things, they just seem to, like, there is no one answer. It's more so a complex combination of all the different factors.
Yeah, absolutely right. Everything about stem cell development relies on everything we talked about with AI and omics and everything else. And they're all, if you're going to put this into people, you got to deal with profits and costs and the business and
Right.
the economics of it all. Yeah, you're absolutely right. Everything depends on everything else.
Awesome. Well, I think we're running up on the time, so we can finish up here. But I really appreciate the conversation, and getting to learn, and especially in a field that I'm not so well versed in myself, has been tremendously valuable for me. And yeah, it's great to have the conversation.
Yeah, Kyle, thank you very much. Thank you for inviting me. It's been a pleasure. Very nice conversation. Thank you.